Hello everyone. In the last video, we discussed about the modification of Mendel's monohybrid ratio in incomplete dominance, codominance and lethal alleles. Today, we are going to discuss about the modification of the dihybrid ratio. Mendel's dihybrid ratio is 9:3:3:1. But in many cases, it is seen that there is the deviation from this dihybrid ratio, or there is the modification in this dihybrid ratio, and this is possible due to the intragenic or intraallelic interaction. This intraallelic or intragenic interaction is known as gene interaction. What is gene interaction? Earlier, according to Mendel, gene function independently and one gene is responsible for a single trait. But later it was found that many different genes together contribute for a single trait in an organism. When two or more genes that are present on the different locus, that is, they are non-alleles, are responsible for a single trait in an organism, then it is known as gene interaction. One such gene interaction is seen in epistasis. In this video, we will discuss about two types of epistasis: dominant epistasis, recessive epistasis, or supplementary gene epistasis. In this, one gene masks the expression of the other gene present on the different locus. Gene which masks the expression of the other gene is known as the epistatic gene, and gene whose expression is masked is known as the hypostatic gene. But this type of the interaction is different from the dominant recessive interaction, because in the epistasis the interaction occurs between the non-alleles, that is the alleles of the genes that are present on the different locus, but dominant recessive interaction occurs between the alleles of a gene. There are different types of epistasis in which different dihybrid ratio is seen. We will discuss these epistasis one by one. First is dominant epistasis. Here the dihybrid ratio is 12:3:1. In the dominant epistasis, dominant gene masks the expression of the other dominant gene present on the different locus. Suppose capital P is the dominant allele which masks the expression of the another gene, capital R. So here P will express itself phenotypically, but it will suppress or mask the expression of the capital R. So P is epistatic to R, or capital P is the epistatic gene and capital R is the hypostatic gene. Capital P gene is responsible for the production of purple color, and capital R gene is responsible for the production of the red color. So let us understand this dominant epistasis with the example. Parental generation is crossed. One is homozygous for the dominant P and recessive R. The other is homozygous for recessive P and dominant R. So it has the purple flower color and it has the red flower color. Then these produces gametes, which fuses and produces the F1 generation, which has all purple flower. Here, if we see the genotype, it is heterozygous for the P allele and also heterozygous for the R allele. So why it is purple? Because this dominant P is epistatic to dominant R. So this dominant P or the capital P masks the expression of the capital R. So the red color is not produced and the dominant P expresses itself phenotypically. Hence all the F1 generation is purple. Then this F1 generation is self-fertilized. All the male and the female plants produces 4 types of the gametes each. And we get the offspring in which 12 are purple, 3 is red and 1 is white. So this 12:3:1 is the phenotypic ratio. Now let us discuss about this ratio, or let us see the explanation of this ratio. When the dominant allele P is present, then it codes for the functional enzyme P. In the presence of the dominant allele R, it encodes for the functional enzyme R. Both the enzyme works on the white substrate. Enzyme P converts the white substrate into purple product and enzyme R converts this white substrate into the red product. In the genotypes in which both the dominant allele P and dominant allele R is present, we get purple color flowers. Like in the F1 generation, all flowers are purple. Why we get purple flower and not the red flowers? This is because the enzyme P and R competes for the white substrate, but the enzyme P has greater affinity for this white substrate compared to the enzyme R. So if both the enzyme P and the enzyme R are present, then this enzyme P will converts all the white substrate into purple product, and hence we get all purple flowers. So in this, if we see these genotypes, at least one dominant allele P is present. Hence functional enzyme P is present and white substrate is converted into purple product. So we get all the plants having the purple flowers, and the number is 12. If we see these genotypes, here the dominant allele P is absent, or there is the lack of the dominant allele P. In the absence of the dominant allele P, enzyme P is not functional, but here at least one dominant allele R is present. So in both the cases, enzyme R is functional. In the absence of this enzyme P, now enzyme R works effectively on this white substrate and converts it into the red product. So in this case, the plant that we get has the red flowers, and here the number is 3. If we see this genotype, it has both homozygous recessive P and homozygous recessive R. So here both the dominant P and dominant R is absent. So there is no functional enzyme P or functional enzyme R. Hence this white substrate is not converted into any color product. So the plant that we get has white flowers. And here the number is 1. So in this way we get 12 purple, 3 red and 1 white flower color plant. So the phenotypic ratio is 12:3:1. Now recessive epistasis or supplementary genes. When recessive alleles present on one gene locus in the homozygous condition mask the expression of non-alleles, that is genes which are present on the another locus, then it is known as the recessive epistasis. So the hypostatic gene expresses itself only when the epistatic gene is dominant. either in the homozygous or the heterozygous state. So let us understand this. This is the parental generation. One red flower plant is crossed with the white flower plant. Here the red flower plant has homozygous dominant R and homozygous recessive P. White flower plant has homozygous recessive R and homozygous dominant P. F1 generation which we get is all purple. Then this F1 generation is self-fertilized and we get F2 generation. This F2 generation has the ratio 9 purple flower plant is to 3 red flower plant and is to 4 white flower plant. So how it is possible? White substance which is present acts as the substrate for the enzyme R. This enzyme R is encoded by the dominant R gene. This then acts on the or works on the white substance and converts it into the red product. This red product, in the presence of the functional enzyme P encoded by dominant P gene, is converted into the purple product. So here we can say that this dominant R is independent. That means it expresses itself. It shows its phenotypic expression, that is the red color, either in the presence of the dominant P or in the absence of this dominant P. But P, it produces its effect, or it expresses itself, or it expresses the purple color in the presence of the dominant R. Because when enzyme R converts the white substance into red product, then this red product acts as the substrate for the functional enzyme P, and this enzyme P then converts this red product into purple product. Now let us see the genotypes of the F2 generation. Here the genotypes which has at least one dominant R and one dominant P gives the purple flower color, because here both the functional enzyme R and functional enzyme P is present. So the white substance is ultimately converted into the purple product. So we get 9 purple flower plant. In this type of the genotype, where at least one copy of dominant R is present, but homozygous recessive P is present. So only functional enzyme R is present, but there is the absence of functional enzyme P. So this white substance is converted into the red product, but it is not further converted into purple product in the absence of functional enzyme P. So in this type of the genotypes, we get red flower plant and its number is 3. Now in the third type of the genotype, where the homozygous recessive R is present, or at least one copy of the dominant P is present. So here this enzyme R is non-functional, but enzyme P is functional. In the absence of this functional enzyme R, the white substance is not converted into red product. So if the red product is not present, then there is no substrate for the functional enzyme P. So the purple product or the purple color is not produced, as a result of which plant has white flower instead of the purple flower. In the fourth type of the genotype, both the R is in homozygous recessive condition and P is also in the homozygous recessive condition. So there is no functional enzyme R or P. Hence, no pigment is produced or white substance is not converted into any color. Hence, the plant gives white flower instead of red or purple flower. So if we see the total, we get white flower plant, that is 4 in number. And this explains the F2 ratio 9 purple is to 3 red is to 4 white. If we see this genotype, here gene R is present in recessive homozygous condition and gene P has at least one dominant allele. And the plant we get has white flower color because in the presence of the recessive homozygous R gene, enzyme R is not present, so white substrate is not converted into red product. So even in the presence of enzyme P, purple product is not formed in the absence of this red product. In this way, the expression of dominant P allele is masked, and this is masked due to the presence of this homozygous recessive R allele. So recessive R gene is known as the recessive epistatic gene. Also, dominant R gene expresses itself independently, but dominant P gene does not express itself independently, but it requires the presence of the R gene for its phenotypic expression, that is the purple color. Hence, R and P genes are known as supplementary genes. Now let us see the comparison between dominance and epistasis. Dominance is allelic suppression. Epistasis is non-allelic suppression. Dominance involves single pair of alleles. Epistasis involves two pair of alleles. In dominance, gene suppresses expression of its allele. In epistasis, gene suppresses expression of non-alleles. In dominance, effect of recessive allele is suppressed. In epistasis, effect of dominant or recessive allele is suppressed. Dominance is due to dominant allele. This dominant allele masks the expression of recessive allele. Epistasis is due to dominant and recessive allele. So this is all for today's video. In the next video, we will discuss about duplicate gene and modification of Mendel's dihybrid ratio. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.